Section 5.3 is on reproduction and meiosis. So I have a little bit of notes for you since I'm not here, but I'm kind of here. At least I was here. This is what we should be able to do by the end of this. Compare and contrast asexual and sexual reproduction. Give an overview of sexual reproduction and outline the phases of meiosis, which is different than mitosis, and explain why sexual reproduction leads to variation in offspring. What we're not going to do is the last thing, define life cycle, identify different types. That gets into a little more detail than I think we need to go into. And I'll tell you at the end which parts of the section we can go ahead and skip. So first is asexual. So A means without sexual reproduction. And you're only going to have one parent, which is kind of weird. You're essentially cloning. Only one parent, and your offspring is genetically identical to that parent. This is done a lot with smaller organisms, bacteria, protists, even some types of animals, but usually more, they're smaller or simpler animals, and some plants will do this as well. The first type is binary fission, which is shown up here with probably what would be a bacteria or a archaea, where it's the simplest kind. It splits into two. The chromosome duplicates, so now there's two chromosomes. Cytokinesis happens, where the cell will fold in on itself, and then you've got two identical cells. They're called daughter cells. This is the parent cell. These two daughter cells should be identical to each other, as well as identical to the original cell. Fragmentation is kind of weird, where a piece of the parent can make a new organism completely. And this is, for these creatures, this is not the only way they can reproduce, but they can reproduce this way. So a planaria is a kind of, or planarian, I'm sorry, is a kind of flatworm. And if you cut it in half, the head will regrow a tail, the tail will grow a head. You end up with two new planaria, which is weird. Hydra, which is shown twice on here, is related to jellyfish. It is a very simple type of animal. Same thing, if you cut a hydra in half, the top will make a new bottom, the bottom will make a new top. And lastly, a sea star. Not really a fish. Sea star is the more accepted name. You can break off a arm, and not only will the original starfish grow a new arm, but the arm will grow a whole new starfish. There are stories of sailors trying to get rid of starfish around a dock, cutting them up, throwing them in the water, and wondering why there are so many more. Well, this is why. They're growing in a new starfish. And budding, where on the parent, a offspring starts to form, gets bigger, and then goes away. The hydra, again, can do budding, where it starts to develop a type of offspring on it, and once it's mature, it releases it, and you've got a second hydra. This is yeast. Yeast can reproduce sexually, but usually reproduces by budding, asexually, by itself. So you can see that some of these yeast cells have another little bud starting. They're going to grow till they're the size of the original cell and break away and become a new cell. There are some definite benefits to reproducing this way. One, asexual reproduction is very fast. The fastest reproducing bacteria that we're aware of can reproduce every 10 to 20 minutes, maybe closer to that 10 minutes. So if you were to think about that, if you were to start with one bacteria, then you end up with 2, then 4, then 8, then 16. Then we're going, what would it be? 64, 128, 256, and it can, every time it gets bigger and bigger, and you start to have logistic growth where it increases exponentially. If you started with 100 bacteria and it divided at that speed, you could have millions in a few hours, which is pretty messed up. You won't necessarily, because it have to have enough resources, it have to have enough space, have to have enough food, have to get rid of waste. But the point is, it can spread quickly, they can reproduce very quickly, and that is an advantage. There's going to be disadvantages as well later, but it's a definite advantage is the speed in which they can reproduce. Sexual reproduction is going to have 2 parents, and each parent is going to produce what's called a gamete. A gamete is a special cell. It is unlike the other cells in the body. For instance, all the other cells in the mom, besides the egg, all the other cells are going to have 2 sets of chromosomes. One from her mom, one from her dad. Same with this guy. All of the cells in his body, besides these, are going to have 2 pairs of chromosomes, one from his dad and one from his mom. And they're going to do something called meiosis, where some of her dad chromosomes and some of her mom chromosomes are going to end up in an egg. But that egg is not going to have 2 sets. It's going to be 1 set. Some of the genetics that this guy's cells have, some from his mom, some from his dad, are going to end up in sperm, but only 1 set. So that 1 set will have a mixture of mom and dad stuff. But it won't have chromosome number 1 from mom, chromosome number 1 from dad. It'll have chromosome number 1 from either mom or dad, chromosome number 2 from either mom or dad, etc. So we go from having 2 pair in these people, total of 46, to having 1 set. I should say 2 sets, not 2 pair. 2 sets in all the adult cells. 2 sets of 23, that adds up to 46. And then 1 set in each of the gametes, being 23. Ultimately, those are going to come back together. You're going to be back to pairs. That's going to develop in a person, and that person's going to be able to reproduce. Hakuna Matata, circle of life. When those gametes do come together, they're referred to as fertilization. And one thing I need to go back and say is, when we are talking about 1 set of chromosomes, it's called haploid. Ploidy refers to how many sets. Haploid is 1 set. We'll represent that often with the letter N. So we've got 2 gametes. Both of them have only 1 set. Our female egg has 1 set of chromosomes. In this case, we just have 1 in here for simplicity's sake. The male has 1 set. They are both haploid. When they combine, you now have what's called a zygote. The zygote is the first cell that has 2 sets of chromosomes. A set from the sperm, a set from the egg. So now we're back up to diploid, or 2N, which is the normal condition for people. All of our cells are diploid, except for, ladies, the egg, and guys, the sperm. So this zygote, this single cell that is diploid, has 2 sets, will divide, grow into a fetus, into a baby that is born, that will eventually be an adult. But somehow you have to get from diploid, 2 sets, down to haploid, 1 set, when those adults get to a reproductive age. So we did mitosis before, which was just cell division, going from diploid to diploid, from 2N to 2N. Now we have to go through meiosis, and it is a little more complicated than mitosis. It is chromosome reduction division. We're going from 2 sets down to 1 set. One diploid cell, or a 2N cell, with 2 sets of chromosomes, becomes 4 genetically different from each other haploid cells. 1 set, represented by N. So we're going to start with this one, and we've got maybe chromosome number 1 from dad, number 1 from mom, number 2 from dad, number 2 from mom. They're going to go through a process similar to mitosis, not exactly, and then they're going to sort of go through another one. But you end up with 4 cells that are different from each other. Each one of these could be an egg or a sperm, and each one is different. And that genetic difference is going to be important for variation so that you can survive your environment. So meiosis is broken down into 2 parts, meiosis 1 and meiosis 2. Meiosis 1 has 4 stages, just like mitosis did. Meiosis 2 has 4 stages, just like mitosis did. During meiosis 1, you've got a diploid cell that remains diploid until the very end. And meiosis 2, you have now a haploid cell that is going to split further. So, starting with meiosis 1, is prophase 1 similar to prophase in mitosis? A few things are going to be the same. Nuclear envelope is going to start to break down. That is the same. Chromosomes are going to be going from spread out chromatin to condensed chromosomes. So now that they are visible, your centrioles are going to move to opposite ends. 2 things that are new though. Homologous chromosomes are going to pair up. The chromosome number 1 from mom and the chromosome number 1 from dad are going to pair up and find each other. The chromosome number 2 from mom and the chromosome number 2 from dad are going to pair up. They're going to find each other. And a weird thing called crossing over will occur, where a chunk of the mom chromosome and a chunk of the dad chromosome are going to swap here and here. It's going to lead to more variation. Metaphase 1, similar. Again, it's going to have to do with the middle, but the difference is they paired homologous chromosomes line up in the middle. With mitosis, it was an X, another X, another X, another X, another X in one line. Now we've got side by side. Here's an X, there's an X. Maybe this is number 1 from mom, next to number 1 from dad. Number 2 from mom, next to number 2 from dad. It's a little different. Paired homologous chromosomes. They're similar, but not exactly the same. They're going to line up in the middle. Those spindle fibers are going to attach to the centromeres on those so that they can move them. Anaphase 1 is going to have movement. Those spindle fibers are going to shorten, and it's going to separate homologous pairs. In mitosis, it was separating sister chromatids. Here in meiosis 1, we are separating homologous pairs, separating chromosome number 1 from mom from chromosome number 1 from dad, and chromosome number 2 from mom from chromosome number 2 from dad. So they're going towards each end. By the way, you could end up with mom on this side, dad on that side for number 1, and dad on this side and mom on that side for number 2. It's completely random as to how they line up. Will it be mom and dad, or with mom on the left, dad on the right? Could be mom on the left, dad on the right. Could be mom on the right, dad on the left. I'll get back to that too. But chromosome number 1 from one of the parents on one end, the other parent to the other end. Telophase 1, telophase 1, I say it both ways. In cytokinesis, they both sort of happen at the same time. While telophase is happening, cytokinesis is also starting to happen. So the spindle fibers that pulled them will break down. You start to reform nuclear membranes around these. The cell is going to start to pinch off and divide. And at this point, we now have two haploid daughter cells because this cell might have chromosome number 1 from mom and chromosome number 2 from dad. It just has one set. It doesn't have number 1 from dad. It doesn't have number 2 from mom. This cell might have chromosome number 1 from dad, chromosome number 2 from mom. It does not have chromosome number 1 from mom. It does not have chromosome number 2 from dad. It just has one set instead of two, so it is haploid. And then cytokinesis is cell splitting, so we're going to split this, and we're going to have two daughter cells that are now haploid with one set. And then it sort of starts over again. I forgot to put a picture on here. Oh no. Anywho, prophase 2 is going to look almost identical to prophase and meiosis, where you're going to have X's next to each other, and they're going to line up in the middle. Not X's side by side, X's in a straight line. Oh, I really wish I had a picture. Those centrioles are going to move to opposite ends. The nucleus that surrounds them will break down, and they will get, I guess I shouldn't say they'll be towards the middle yet. There will be, though. So really, these are just about in prophase 1 once this separates, if you need a visual. Oh, there it is. Huh. I outsmarted myself again. Metaphase 2. I said prophase 2, right? Yep, metaphase 2. Now this is going to look very, very similar to metaphase in mitosis. They're going to line up in the middle, those X's, and they're going to get pulled apart. Spindle fiber is going to attach. They're going to pull them apart. They're now pulling apart sister chromatids. If this was number 1 from mom, the two copies of chromosome number 1 from mom are going to get pulled apart. The two copies of chromosome number 1 from dad are going to get pulled apart. The two copies of chromosome number 2 from dad are going to get pulled apart. The two copies of chromosome number 2 from mom are going to get pulled apart. Now, they're not exact copies because there's little pieces that have switched, but they're pretty close. Anaphase 2 is going to look similar. Those spindle fibers are going to shorten, so the sister chromatids are going to separate. One from mom, one from mom, one from dad, one from dad, two from dad, two from dad, one from mom, one from mom. Telophase, ooh, this should say telophase 2. Also my bad. Telophase 2 and cytokinesis. Pretend that's a 2. The spindle fibers are going to break down. Nuclear membranes are going to reform. The cell's going to go through cytokinesis and divide. And now we end up with 4 haploid, meaning one set, daughter cells. And each one is going to be different than the other. For instance, if this was number 1 from mom, but with all mom stuff, number 2 from dad with all dad stuff. This also was number 1 from mom, but it has pieces of dad DNA on there. And this was number 2 from dad, but it's got a little piece of mom DNA on there. They're different. This was number 1 from dad, but it has little chunks of mom DNA on it. This was number 2 from mom with a little chunk of dad DNA on it. This was number 1 from dad, number 2 from mom. Each one unique, each one different. I have a video. Hopefully it works. It's like Inception. It's a video within a video. Unless it didn't work there. Ooh, let's try this.
The cell prepares for meiosis just as it does for mitosis. But as the DNA condenses,
I'm not sure if you guys got the audio or not. If you didn't, my bad. I didn't know what would happen when I did Inception. I will probably post that video separately just in case that audio did not work. So once you're at the end of meiosis, you now have 4 haploid cells. And those cells are going to become gametes. If you're a guy, those 4 haploid cells are going to become 4 sperm. If you're a girl, one of those 4 cells is going to become the egg, and the other 3 are sort of going to get their cytoplasm stolen away to make the egg bigger. Notice the size difference. The sperm is not nearly as big as the egg. Because of the way this happens with sexual reproduction, we end up with a lot of genetic variation, a lot of variation in genes. And this should make sense because even though you have the same parents, you are not exactly like your brothers or sisters. Because each time you're going to have different variation. So the first kind of variation happened in prophase 1, where 2 homologous pairs line up together and exchange chunks. So maybe mom DNA, dad DNA. Now this mom DNA has a chunk of dad DNA. This dad DNA has a chunk of mom DNA. That's going to be another way to get variation. You're not going to get purely mom or purely dad genetics. The other is independent assortment. When they line up side by side in metaphase 1, it's completely random as to which goes to which side. You might have chromosome 1 from dad to the left and 1 from mom to the right, and 2 from mom to the left and 2 from dad to the right. And if you had that, you would end up here and then end up here. However, it's just as likely that they would line up this way, with 1 from mom on the left, 1 from dad on the right, 2 from mom on the left, 2 from dad on the right. So you end up here and here, completely different combination. So these 2 together lead to a lot of variety, and the fact that pretty much every sperm cell is going to be different, pretty much every egg cell is going to be different. When you combine a random sperm and a random egg, you're going to have more variation. So trying to make sense of this very complicated thing with what we did before. Mitosis was just a way to get more cells. You have a cell with 2 sets of chromosomes, and it copies all of its material and divides. So you still have cells with 2 sets of chromosomes. Meiosis is different. Meiosis, you're going to start with a diploid cell, 2 sets. It's going to make copies, and then it's going to divide in a different way, so that you have 1 set that's duplicated and another set that's duplicated. And they're going to divide further yet to have a single set non-duplicated. Meiosis only happens in order to make gametes. This is confined to the testes and the ovaries. It's the only place it's going to happen for humans. Mitosis happens everywhere else. Your muscles, your bones, your skin, all of that. When you are a baby, mitosis gets you bigger. When you were just conceived, mitosis makes you develop into a fetus, and then a baby, then a kid, and then an adult. So the human life cycle has part of it, the part that we're familiar with, visible people. Your cells are diploid. You have 2 sets. You started as a zygote, a cell with 2 sets of chromosomes, and it divided, divided, divided. You got bigger and bigger and bigger, and you end up being either a boy or girl. However, if that boy and girl love each other very, very much, and they decide they want to have a baby, the boy's testes and the girl's ovaries are always going to be making gametes. Girls are going to be making eggs, boys making sperm. They have to go through meiosis to get there, to go from 2 sets in the guy down to 1 set in the sperm. 2 sets in the girl down to 1 set in the egg. So now they are haploid. They have 1 set. But when a haploid sperm and a haploid egg combine, that's called fertilization, you're back up to 2 sets. New baby grows up, whole thing happens again. Circle of life, Hakuna Matata. So within that section, it gets even more in depth, and I'd say this is plenty. So you can skip these sections. The section that says sexual reproduction and life cycles, don't worry about it. Haploid life cycle, don't worry about it. Diploid life cycle, don't worry about it. Alternation of generations, don't worry about it. So hopefully we were able to do 3 of these things, compare and contrast asexual and sexual. How are they the same? How are they different? Give an overview of sexual and go through the phases of meiosis, outline them, and explain why sexual reproduction leads to more variation. Remember, you can always replay this. I'll put it on Classroom. And please, 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 please read the section except for those. And keep in mind there's going to be not a ton, but quite a few vocabulary words.